Good morning to everybody. I would like to talk about the role of tight junctions, the role of tight junctions on the intestinal integrity. My presentation will be a short presentation because we will have time to discuss deeper during the coffee break, if you want. So, as you know, the intestine has two critical functions that ensure the intestinal health. First, one of the main functions of the intestine is to allow the passage, the transport of nutrients, electrolytes, water from the lumen into the circulation. But also, the intestine has to restrict the passage of pathogens, antigens, and also the harmful substrates. So, the maintenance of these two critical functions are named the intestinal barrier function. In poultry production, some ingredients and additives improve the intestinal health and determine this improvement not only in terms of production but also in terms of quality of the products obtained, consumers' safety, and animal welfare. So one of the components of the intestinal barrier is the selectivity of the epithelium. Other components that also contribute to this intestinal barrier are microbiota, the mucous barrier, and the immune system. It's important to notice that these components work all together and they have a really tight relationship between them. Let's talk now about the epithelium permeability. To cross the epithelium, we have two different pathways. The transcellular pathway that is associated with the transport across the epithelial cell, first through the apical membrane and then through the basolateral membrane. We have also another pathway, the paracellular pathway, that is associated with the transport of the different substrates between the cells by the intercellular space. The tight junctions, the most apical junctional complex in the epithelia, are responsible of the regulation of paracellular permeability. Tight junctions are formed by different proteins. We have in the tight junction the transmembrane proteins. These transmembrane proteins join one cell to the neighbor one. And the most studied transmembrane proteins are occludin and also the claudin family. In the tight junction, we also have cytoplasmatic proteins. The most studied are the ZO family that are the nexus between these transmembrane proteins with the other component of the tight junction, that is the cytoskeleton proteins. Also, in the tight junction, there are a lot of regulatory proteins that ensure the function, the correct function of the tight junction. But I have a question for the audience. Which are the functions of tight junction? Which of these answers do you think that is the correct one? A, B, C, D? C? C? The correct one is the D one, because tight junctions not only regulate paracellular permeability, but also tight junctions are able to regulate cellular polarization. because tight junctions are in the interface between the apical domain and the basolateral domain. So this tight junction restrict the movement of proteins from the apical domain to the basolateral domain, with the objective to allow a vectorial transport. Moreover, tight junctions regulate proliferation and differentiation. That is two functions that are very important in epithelial cells. Let's talk about paracellular permeability. Tight junctions determine two pathways. The pore pathway, that is described to be a high capacity and a selective pathway. This pore pathway allows the passage of mainly ions, small molecules, and is regulated mainly by clothings. We have another pathway, is the leak pathway. This pathway is described to be a low capacity and a non-selective pathway that allows the passage of larger molecules and is regulated by occludin, GO1, and the cytoskeleton proteins. These two pathways are not independent because it is described that occludin can also regulate the pore pathway. In in vitro cultures, it is easy to determine paracellular permeability by measuring the transepithelial electrical resistance that inform us about the resistance of the epithelium to ion fluxes. Moreover, we can measure paracellular permeability by determining the paracellular fluxes of some markers specific from the paracellular pathway, like mannitol or some dextrans or the lucifer yellow. I have also a question for you. In an optimal epithelia or barrier function, the transepithelial electrical resistance values are high or low? What do you think? High. Okay, correct. The epithelium resistance to ion fluxes is very high in a normal situation. But when this barrier is disrupted, these values, these tier values, decrease. In contrast, paracellular fluxes of these markers of the paracellular pathway, what do you think? In optimal situation, these fluxes are high or low? Low, perfect. An optimal barrier is characterized by low fluxes of these paracellular markers, but when there is a disruption of this barrier, these fluxes are increased. TIR is not able to discriminate between the pore and the leak pathway, and to discriminate between these two pathways, we have to use some larger paracellular markers with a high molecular weight in order to differentiate between the pore and the leak pathway. So, a disruption of tight junction extractor leads to an increase in paracellular permeability. This increase in paracellular permeability could be by different mechanisms. One of the mechanisms involved in the disruption of tight junction extractor is a decrease in the expression, the genetic and also the protein expression of some tight junction protein. In this sense, some tight junction protein could regulate the gene expression. But also, a disruption of tight junction structure could be due to a delocalization of tight junction proteins from the plasma membrane to the cytosol. Moreover, another way to disrupt the tight junction structure is to produce a contraction of the actomyosin ring. In that case, with a contraction of these proteins, we obtain an opening of the paracellular pathway. So, tight junctions can be regulated in response to both physiological and pathological stimuli. Let's see some examples. One of the well-defined examples of physiological regulation of tight junction is that following the activation of sodium nutrient cotransport. The transport of glucose and some amino acids with sodium leads to activate the myosin light chain kinase. This enzyme is able to produce a contraction of the actomyosin ring and also produce a slight tight junction internalization. So in that case, there is a slight increase in tight junction permeability, which is the objective of that. The objective is to increase water absorption and to increase also the paracellular transport of small nutrients. But mainly tight junctions could be regulated in pathological situations. And one of the actors that are involved in these pathological regulations are cytokines and also oxidative stress. One of the main cytokines that is involved in the regulation of tight junctions is TNF-alpha. This pro-inflammatory cytokine is able to induce also, but in that case, a more pronounced activation of this myosin light chain kinase. In that case, this activation leads to a contraction of the actomyosin ring and also leads to an increase of the contraction of the actomyosin ring and also a pronounced internalization of occludin from the tight junction to the cytoplasm. It has been described that also oxidative stress that could be produced by different mechanisms could modify the tight junction structure. But it's important to notice that tight junction can be regulated by components of the diet. For example, HMTBA, the hydroxy analog of methionine, is able to partially protect from the disruption of the barrier, from the decrease on TIR values produced by TNF-alpha. In that case, the main results we have obtained is that HMTBA is able to maintain the integrity of occludin in the tight junction. Moreover, HMTBA is also described to partially protect from the disruption caused by hydrogen peroxide, and in that case, by maintaining the structure of GO1. So it's an example of some dietary components that could also regulate tight junctions. Moreover, probiotics are also described to enhance barrier function, to prevent intestinal barrier dysfunction, and mainly they do this function by immunomodulation. We have observed that also prebiotics such as beta-galactomanans are able to help to the microbiota and to the probiotics to change the balance of the different cytokines with the objective to restore the epithelial barrier function after Salmonella colonization. So, to take home, it's important to be sure that tight junctions determine paracellular permeability. These tight junctions can be regulated in response not only to physiological but also to pathological stimuli. And the last important thing is that tight junctions can be regulated by dietary components. So, thank you very much.
Thank you very much for this interesting overview. Other questions?
The first question, how important is the paracellular absorption compared to transcellular depending on the different nutrients? The paracellular permeability is, in optimal situation, very low. So normally, nutrients has to cross the epithelium by the transcellular pathway. But in some times, normally when the nutrients are very high in the lumen, the epithelia could regulate the junction to increase the absorption of this nutrient by the paracellular pathway. Tight junctions can regulate the effect of fasting. Oh, sorry.
The last one.
The last? Tight junctions can regulate the effect of fasting on gut permeability, I think. Yes, tight junction could be regulated depending on the concentration of the different nutrients in the lumen. So we have to keep in mind that this regulation is not only to protect the barrier, but also to increase nutrient absorption.
Okay, thank you very much.